Assalamu alaikum. Today we discuss about the rDNA, that are the recombinant DNA, the recombinant DNA. The recombinant DNA are the DNA that carry some other DNA that are known as the recombinant DNA. So we discuss about the steps of the recombinant DNA, how we get the recombinant DNA, and how we get multiple copies of the this recombinant DNA. So now, first of all, we have this donor cell. This is the donor cell, and in other hand, we have bacterial cell. This is the bacterial cell. In donor cell, we have DNA. This is the DNA. And in bacterial cell, we have plasmid. This is plasmid. Now we use the lysozyme. Lysozyme. This lysozyme from the donor cell and from plasmid, it can act on both cells. When it can act on the donor cell and plasmid, it take out this DNA from the donor cell. We have this DNA from the donor cell and the plasmid. It take out the bacterial cell, the plasmid from the bacterial cell. This is the plasmid. Here we have DNA, and here this is the plasmid. We use the lysozyme and we take out the genetic material from the donor cell DNA, and from the bacterial cell we take out the plasmid. So now we use the restriction enzyme. We use restriction enzyme. When we use restriction enzyme on the donor cell, it acts on this DNA and we get different fragments of this DNA. And from the plasmid, it cut the plasmid from one side. Now we choose from this genetic material, from this DNA, we choose the gene of interest. Gene of interest. This is our gene of interest. We want this gene, and we want the rDNA, recombinant DNA from this gene. So now we use the ligase enzyme. We use ligase enzyme. When we use ligase, it helps the gene of interest bind with this plasmid. This is plasmid, and this is our gene of interest. It can attach, it can bind with the plasmid with the help of the ligase enzyme. Now we have the rDNA, that are the recombinant DNA. This is the recombinant DNA, and now we want different multiple copies of this rDNA. We use the electroporation technique. Electroporation technique. When we use electroporation technique, by using this technique, we insert this rDNA into the E. coli. We insert by the electroporation. this rDNA into the E. coli. And when it replicates, we get different bacteria, E. coli cells that are the recombinant, that have the recombinant DNA. Now we have multiple copies of the recombinant DNA of the E. coli. So this is the process and the steps of the recombinant DNA. If you like this video, subscribe, like and share it. Thank you.